Okay, good morning, everyone. Happy Friday. I hope all of you are somewhere where you're staying nice and dry. I guess that's one of the advantages of online teaching, right? You can stay home wrapped up in a blanket and still attend a lecture. Okay, so let's talk today about the last set of new material that you are responsible for next week's exam. So just as a reminder, here we are at the end of week 8. So today is lecture 19 on the endoplasmic reticulum and Golgi. So this is where we'll wrap up our discussion about different membranes that make up our cells. On Monday is our review session. So during lecture time, we'll do a review. And then Wednesday and either Thursday or Friday is your second exam. So be aware that you do have a homework assignment that will be due next week. The homework assignment is meant to help get you into good shape, discussing or thinking about the different topics that are going to be on the exam, having to do with gene regulation, and also what we've been discussing about membranes. So I know this exam is going to have a lot of tricky stuff on it. It's definitely a step up from the first exam. So it's up to you to use this weekend well and get organized and start reviewing the material. The homework will be due next Friday by midnight, right? So you have some extra time to do it, but don't slip up and forget to turn it in just because it's not your normal time of turning in homework, because I don't want it to be a thing where you end up not doing the homework at all until Friday night after the exam, or you forget to do it, because then that's just kind of silly, right? Because the homework is meant to help you get ready for the exam. It's not meant to be some busy work thing that you still have to remember to just turn in, right? So make the most of it. I believe your TI, some of them will be organizing optional review sessions, so you can keep your eyes open for that as well. Okay. When we've been talking about membranes so far, we've mainly been talking about the cell membrane, right? The boundary between the cell and the interstitial space or fluid surrounding the cell in our tissues. And our discussions about different proteins in the membrane that help move different things back and forth from one side of the membrane to the other. For convenience sake, we've just been sort of pretending like we're talking about the outside of the cell. But of course, the principles of moving things back and forth across membranes applies very much to what's going on inside of the cell as well, and the different types of spaces inside of the cell where certain biological functions are taking place. Remember, life works because biological molecules are being preferentially put together in different places. Put together meaning you're collecting or concentrating certain molecules so that there's a greater chance for biological reactions to happen, compared to if you just simply had everything scattered around and relied upon molecules bumping into each other by chance alone, right? Because I think you all intuitively know that that just wouldn't work for life, because you wouldn't have any control, right, over all these complicated processes. So membranes play an essential role in defining certain spaces within the cell where certain molecular components of life can be concentrated, put together, so that things can favor the interactions that are meant to happen in order for the cell to actually carry out its cellular functions. So not just a membrane on the outside, but lots of membranes on the inside that define things like organelles. Right? So that network of membranes inside of the cell, we call the endomembrane system. So let's look at this. So here's an example of some liver cells, some human hepatocytes. Right? So these are cells in your liver that do things like, for example, store excess sugar in the form of glycogen. And then the glycogen is also broken down in these cells back to glucose, so that it can be released in the blood, so that in between meals, if you haven't eaten anything for like 8 hours, right, your liver is able to release stored up glucose so that the cells in your body can get energy. At low magnification here, it's pretty obvious where each of the outer cell membranes of each of these cells are. And you can see the nucleus, right? So the nucleus, of course, has its own membrane, the nuclear envelope, which is part of the endomembrane system. But you can't really make out what's going on in the cytoplasm, right? It's all this sort of pink speckled stuff. But if you zoomed in and under high magnification, then you could really see, oh, here's the nucleus, right? And you see all kinds of curious squiggly things in here, right? So you have membranes forming vesicles, membranes forming different types of organelle structures, like potentially mitochondria. Then you have lots of squiggly examples of membranes, right? So all of these are playing important functions, and all of these use those principles that we've been discussing about how the cell moves things back and forth across membranes. So let's explore this today. So among these membrane structures are things like the nucleus, of course, right? So the nucleus obviously is an example of how you use a membrane to contain certain macromolecules together. Our DNA, right, is concentrated in our nucleus, and that's where it stays, right? So that's the only way it would work to have our genome protected and accessed so that our genes can be expressed, is if it's protected inside of a particular space where things like RNA polymerases and other proteins like transcription factors are more concentrated and have a chance to work on our chromatin. But other structures like big organelles like mitochondria, things like that, right? They're out in the cytoplasm. And I said there's all sorts of funny squiggly membranes. Examples of those are, for example, our endoplasmic reticulum. Oftentimes, when people talk about the endomembrane system, they favor focusing on structures like the endoplasmic reticulum and the Golgi complex. So both of those we'll talk about today, right? Because these are examples of membranes that are critical in order for the cell to do certain things having to do with the production of proteins, as well as things like detoxifying certain types of poisons that might be part of the cell. So here's the ER. We have studied the ER a little bit, only in the context of understanding that a lot of protein translation and folding happens in the ER. So in this middle cartoon here, you can see the two forms of the ER. We call the rough ER on the left over here, and the smooth ER on the right. Importantly, I like this cartoon a lot because it really highlights the fact that the ER is one continuous, right, highly folded sort of network of caves in a way. Right, so imagine if you have one enclosed space, right, and you just kind of like folded that enclosed space in all kinds of elaborate ways, right, you would get essentially the ER. So the light blue represents the lumen of the ER, or the inside part, if you're inside the cave, if you want to say, right? And then the dark blue represents the actual membrane that defines this space. That's why if you were to cut this in half, cross-section, the ER looks like a whole bunch of just like these tunnels, and you don't appreciate that they're all connected. Right, like this set of tunnels down here doesn't look like they're connected to this set of tunnels up here if you were just to look at it in cross section. But in this a little bit more three-dimensionally drawn thing, you can see how they're actually part of the same space. On the on the left is a electron micrograph of the rough ER. Right, and you see it's called rough ER because it's studded with all these little black dots in this picture here, which are the little green dots in the cartoon in the middle. What are those? Those are the ribosomes, right? Remember when ribosomes in the cytoplasm have a chance to assemble on mRNAs that are coming out of the nucleus, and the ribosomes are beginning to translate those mRNAs into a chain of polypeptides that represent the new protein being made. Many proteins require that the ribosome ends up sort of docking to the wall of the ER, the rough ER, so that the translation of the peptide chain finishes inside the ER so that the cell has a chance to modify the protein. Right? So when you look at this on the left, the rough ER, you can really appreciate, right? The wall of the rough ER is just lined with ribosomes, busy finishing the translation of proteins into the rough ER lumen. On the right is smooth ER. Notice again, it's all continuous with the rough ER, but here you don't see any ribosomes, right? So here's the cross section of the smooth ER, and you can really see it's just a whole bunch of little spaces, right? Little tunnels. So you can see here the cartoon, that's how it looked, these light blue sort of little dots, right? That's what you see over here, right? But yet it's all a continuous space. In terms of functions of the rough ER versus smooth ER, okay, well, I mean, we've already talked mainly about the rough ER. So let's just quickly in one slide, just blow through this part and review. Remember, the rough ER is mostly responsible for protein synthesis and also protein processing. So it's in charge of making sure that proteins that are being made by ribosomes that are being co-translationally imported into the ER, they fold properly. Also, if you have proteins in the cytoplasm of your cell that are old or damaged and they're no longer folded properly and they need to be gotten rid of and recycled, right? We have cellular recycling machinery that breaks down misfolded proteins and sends the short little peptide fragments into the ER, right? So that they can be processed and recycled, removed. Importantly, in the rough ER is where proteins get finished, not only folded, but also having different post-translational modifications added to the proteins. Right? Things like small little groups, like phosphate groups, acetyl groups, things like that, or even larger groups, like sugar chains. Right? So many proteins are glycoproteins. They have these carbohydrate chains linked to them. Right? And importantly, if you have different proteins that are being finished in the ER, and if some of those proteins are meant to work together as larger complexes, right? Now, we've certainly looked at a number of different examples of big complexes. The very last slide of the last lecture, I showed you that remarkable complex, the V-type proton pumps, which are ATPases that move protons from one side of a membrane to another, right? That's the thing that's shaped like a gumball machine made up of all these wonderful separate proteins, right? That have been made separately and finished in the ER, and the inside of the ER is a space for them to come together to form the gumball machine that then can be moved out and ready to work. Okay. So let's look on the right here. This is all stuff we've learned before, right? The ribosomes are coming together on the mRNA, and as they begin to put together amino acids that are coming in with tRNAs carrying different amino acids into the ribosome, and they get strung together. Remember, we said as the emerging peptide chain is coming out, there are certain amino acid sequences that serve as kind of like a signal. In other words, there are proteins that would be floating around here that aren't shown that would bind to this signaling sequence and then bring or sort of pull the ribosome along with the mRNA to the wall of the rough ER, right? And that's why this ribosome would now be one of those many little dots studding the outside of the ER, like shown on the left, right? And then it would finish making the rest of the peptide chain into the ER. And as part of that, the signal sequence, whatever that first few amino acids were that took care of getting this translational process moved to the ER, that might just be cut off, right? Because that's no longer actually part of the finished protein that's going to be made. Right? So this light pink signal sequence, I'll just say, you know, it's very important. Our cells use these. And in fact, as an example of how kind of neat it is to understand how these work, the mRNA COVID vaccines made by Pfizer and Moderna, right, as part of the way they're engineered, they are mRNAs that code for the particular protein that comes from the virus that you want your immune system to recognize, right? And as part of the vaccine, that mRNA that's engineered, they put in a signal sequence in there, right? A signal sequence that has been optimized so that when that gets translated as part of the viral protein that's being made, right, it does a tremendously effective job of getting your cells to bring the ribosome to the ER so that your ER can make high levels of this viral protein to maximize the ability of your immune system to be able to see it, right? So that's all a function of this signal sequence type of mechanism. All right, so that's kind of it with the rough ER, So that's pretty much stuff that you already hopefully knew. But what about the smooth ER then? Right. So again, here you look at this cartoon. Here's the nucleus, the nuclear envelope. You can see it is continuous with the rough ER. Right? The space of the membranes just kind of all connect together. And then you have the rough ER with all the little blue dots are the ribosomes. And then you have this part. This is the smooth ER. Right? What does the smooth ER do? Here, look over here, you can see this is a nice picture, right? You can see the transition from the rough ER on the right to the smooth ER. So the smooth ER is in charge not really of dealing with proteins, because that's sort of the job of the rough ER. Smooth ER carries out important vital functions of detoxifying certain types of organic and as well as potentially inorganic compounds that might have entered your cell. And if they're not dealt with, they might act as poisons, right? They may end up binding to different things and interfering with the normal functions of cells, right? The smooth ER is also where glycogen metabolism takes place. We mentioned liver cells on an early slide and how they store glycogen, right? So when your body needs it, it is the smooth ER inside of your liver cells that are then in charge of breaking down the glycogen back into glucose molecules. The smooth ER is also a place of storing certain ions, importantly, things like calcium. Certain tissues of your body utilize high levels of calcium in order to perform their functions. For example, our skeletal muscles, right? The ability of our muscle cells to move depends upon having high levels of calcium available. And the smooth ER is important for the manufacturing of certain non-proteins, but still very important molecules like signaling molecules such as steroids, right? Steroids are made in the smooth ER. So let's take a look at these one by one. Detoxification. So many poisons or medications or even carcinogens, things that cause cancer, are small hydrophobic molecules. So the smooth ER, the wall of the smooth ER, has a family of proteins in the wall called the cytochrome P450s. So these CYP proteins, or CYP, they have a heme group. A heme group is a little structure that's particularly good at binding oxygen. So you're like, heme group, you mean like hemoglobin? Yeah, right? So the point is this heme group is used in your cells for different things. In your red blood cells, there's hemoglobin, so a protein that has a heme group like this that it's holding onto that helps it carry oxygen. Here in the smooth ER, you have these cytochrome P450 proteins. So here, this squiggly thing is the cytochrome P450. Part of its tail is hydrophobic, so that's how it stays kind of anchored to the wall of the ER. Another part of the protein then has a part that can bind a heme group, right? So these CYP proteins work by transferring a hydroxyl group onto target molecules like small hydrophobic molecules, making them hydrophilic and therefore easier for your kidneys to be able to flush them out. What do I mean by that? Right? Remember, kidney cells, like the cells of your nephrons in your kidneys, are in charge of constantly filtering tons and tons of water, right, every single day. And in that water, hopefully, are different things that you want to expel from your body, right? So to be able to have things like a drug, a medication, or a poison, right, removed from your body into your urine, then intuitively, the better that this substance can be carried by water and handled by things like the aquaporins of your nephron cells, right? The more efficiently your body can eliminate them. So the cytochrome P450s in the smooth ER take care of making these compounds more hydrophilic. Let's look at an example here. So here's phenobarbital. Right? Phenobarbital is a depressant. It's a sedative. It's often used in modest amounts to be a sleeping aid. Right? So phenobarbital consists of this barbituric acid ring. And if you look at this ring here, you'll see that it has amine groups. So you say, we've seen other things in this class that are small rings with amine groups, right? Like what? Well, like this could be a nitrogenous base. right? If this was added and used to make something like a nucleotide, right? If you were to bind this to a sugar and a phosphate group, but you're like, wait, do we use this as a nucleotide, right? To make DNA or RNA? No, right? Because our bodies, we don't. But it doesn't mean that compounds like this exist. And that's why, right, in your body, if you had this compound, it would react in different ways, right, with other types of proteins. So linked to a phenol group. This is a phenol ring, so this makes phenobarbital. Right, so phenobarbital works as a central nervous system depressant because when it binds to different proteins in the membrane of cells, especially your neurons, it depolarizes the cells. In other words, it opens up certain ion channels like chloride ion channels, Cl minus, and messes up your neuron's ability to actually on purpose move chloride ions back and forth to set up gradients. Right? Remember, we talked about the importance of cells setting up intentional imbalances of ions so that the cell can then take advantage when the time is right of letting things flow down concentration gradients. So chloride concentration gradients are very important for the functioning of your neurons, the ability of neurons to send signals back and forth by depolarizing or allowing ions to flow down their concentration gradient. So phenobarbital messes up your ability of your neurons to actively keep this chloride disequilibrium. And so as a result, your neurons of your central nervous system, they lose their sensitivity, right? And this also contributes to things like sleepiness. So when you have phenobarbitals in your system, the cytochrome P450s on your smooth endoplasmic reticular cells, they go to work. They add hydroxyl groups to them, and it makes your kidney cells have a much easier time then getting the phenobarbital out of your system into your urine. So if you take more and more phenobarbital drugs like this, what happens is your cells, right, they realize, hey, you know, we're getting more and more of this drug building up and we got to get rid of it. So we better actually have more cytochrome P450 proteins go to work. So your cells will actually upregulate the production of your cytochrome P450 genes. You will make more of the cytochrome protein. Your smooth endoplasmic reticulum inside of your cells will get larger and larger to be able to handle having more of these cytochrome P450s on them. And what does that mean? Over time, as your body gets better and better of quickly getting rid of drugs like this, you need to take higher and higher doses of the drug in order to still maintain the level of that drug functioning in your body. So that tolerance that you build up is a big part of drug addiction, right? And so a lot of times drug addiction is just a function of the cells of your body upregulating the genes that are in charge of making proteins that eliminate those drugs, so that evolutionarily, right, this is a way that our cells protect themselves from being poisoned. Okay, another example. The smooth ER involved in glycogen metabolism. So we already learned that glycogen is a great example of a long polysaccharide of glucose molecules that's put together in our cells as a short-term way of storing extra sugar. Right? Because again, evolutionarily, if you have a large meal, right, and you have lots of sugar in your blood, way more sugar than your body needs at a time, evolutionarily, you can't count on the next time you're going to have a nice meal again, right? So you don't want to waste the sugar. You want to hold on to it. So we string it together as glycogen, and that happens inside the liver cells, the hepatocytes. So this is a zoomed in portion of a hepatocyte. You can see what are these dark kind of, sort of like fuzzy looking things. These are glycogen granules, right? So each one of these is a long, intricate set of glucose molecules chained together. And look, what are these little squiggly, bubbly things that are sort of interspersed here? Hey, that's your smooth ER, right? So on this side, the smooth ER is where these glycogen granules are. On the right is your rough ER. So you can see the little black dots that are the ribosomes again. So when the time is needed for your liver cells to break down the glycogen to get that individual glucose molecules out of it. That's done by the smooth ER. Here, let's see, you have a glycogen granule, right? So this thing would be one of these big fluffy looking glycogen granules. If you were to kind of like tease out part of it, you would see it's a chain of glucose molecules. So we have proteins that phosphorylate the individual glucose. So we have a glycogen phosphorylase that transfers a phosphate group onto the carbon 1 position of one of these glucose monomers, like this one here, right? And as a result, it breaks it off of the glycogen chain, right? Then we have another protein that basically moves that phosphate group onto a different carbon position, to the 6th carbon position, right? And this now puts it in the right shape so that the wall of the smooth ER, you have a protein called glucose 6-phosphatase, right? You're like, I hate these words, these names, they're so confusing, I can't memorize them. Well, then don't memorize them. Actually understand what the name means, right? And if you understand it, then it's not really just rote memorization. Glucose 6-phosphate, because it's glucose with a phosphate on the 6th carbon. Then you have a protein that works on glucose 6-phosphate. And what it does is it breaks the phosphate off. So it's called glucose 6-phosphatase, this protein. It breaks the phosphate off, and then you have your free individual naked glucose molecule that then can be transported across the membrane. So again, the importance of membrane transporters, and put into the blood, where it can then flow to somewhere else in your body, away from the liver, somewhere else where tissues need to take this glucose up, right? And maybe those other cells take up the glucose using a transporter like the GLUT1 transporter, right, that we've been learning about. All right, I mentioned that certain cells, their smooth ER works by storing certain ions like calcium. So I'm not going to go into the physiology of skeletal muscle cells too much. This is something that those of you who end up taking different types of animal physiology classes, you'll look at more extensively. But let's just say that here we have a muscle fiber. And if you were to look at really closely, they're made up of individual muscle cells, right? And these skeletal muscle cells, here it is, this fiber in cross section. You would have a nerve that comes and is meant to send the electrical impulses that tell this muscle fiber to contract. If you look at where the nerve, so this is the end of the nerve here, synapsing or pressed up against the muscle cell, which is down here, right? That nerve is going to release certain types of signaling molecules like neurotransmitters that are received by the muscle cell, that causes the cell membrane to depolarize. Again, another example of a cell making use of the fact that it had intentionally been working hard to maintain a charge disequilibrium across the membrane, plus on one side, minus on the other, just so that when the time is right, it could allow that charge to depolarize and something could happen. And that depolarization causes the special smooth endoplasmic reticulum of muscle cells, which we give the special name sarcoplasmic reticulum, but it is just the smooth ER of these muscle cells, to release high storages of calcium ions. And these calcium ions then go to stimulate the contraction of actin microfilaments inside of your muscle cell, causing the cell to contract. Okay, so don't get bogged down by the physiology of muscles here. That's not what this is about. This is just showing you another example of why smooth ER actually is really important inside of your cells. And lastly, the example I wanted to give is that the smooth ER is involved in the production of different types of signaling molecules like steroids. So we said before that steroids come from the parent molecule, the starting molecule is cholesterol. And we said cholesterol is abundant in the cell membrane, right? Not just the cell membrane on the outside of the cell. Hopefully you realize now that all of these endomembrane membranes inside of the cell are made the same way, right? They are phospholipid bilayers with various proteins in them, right? So really not that much different than the cell membrane on the outside, except for which particular types of proteins are part of the membrane of something like the nuclear envelope or a mitochondria, for example, compared to what proteins are meant to be part of the outer cell membrane. But cholesterol, your cell has a lot of it, right? Because it's a part of the cell membrane to start with. And that's the starting point for making certain types of steroids, like the sex hormones. Right? So we mentioned before that things like androgens in males, like testosterone, and estrogens in females, right? They come from cholesterol. Right? So your smooth ER is in charge of making things like testosterone and estrogen. So here's cholesterol, right, as you'd find it in the membrane. And let's see what testosterone looks like. All right, so this is testosterone. And right away, hopefully you're like, oh, I totally see how it is that testosterone is made from cholesterol, right? You've basically removed this, this hydrocarbon chain up here, right? And it's like, okay, then you have a hydroxyl group over here instead. And now you have an oxygen here double bonded instead of a hydroxyl group here, right? And that is testosterone, right? So if that's testosterone, which is in charge of male reproductive system anatomy, other primary types of male features, as well as secondary, right? Everything from hair growth to musculature to like a deep voice, things like that, are all these sort of secondary factors as a result of testosterone signaling. Then what about in the female body, right? What does estrogen look like? You must think, well, estrogen must look pretty different, right? So that you don't end up triggering testosterone effects. Well, here's estrogen, right? It's practically the same thing, right? In fact, what is the difference here? Testosterone has a methyl group over here, right? That has been removed in the estrogen, right? And estrogen still has the hydroxyl group over here that has been removed in the testosterone, right? So the amazing thing about this is that testosterone versus estrogen, right? Even if you don't know a lot about the endocrine system, hormone system, hey, we all know testosterone, estrogen is responsible for the differences that you see between men and women, right? Our physiology, our biology. But yet, how can it be, right, that these molecules that determine that are so similar in structure? This is a nice reminder, once again, right, that we're talking about molecular biology and how these molecules are going to be received by different proteins in different cells that are responsive to testosterone versus estrogen, right? That the ever so subtle differences in shape of the estrogen versus testosterone means that those proteins that bind to them have to be ever subtly so different in shape, right, in order for the proper interactions to happen, right? Which I think is kind of remarkable. All right. So that is our discussion of the ER. Let's move on to the Golgi apparatus. So when proteins are made in the rough ER and they are modified and folded, and maybe different proteins come together inside the rough ER to be assembled into a complex that's meant to work together now, right? How does that end up actually being put or sent to the right part of the cell where, this machinery is meant to work, right? That's the job of the Golgi. So the Golgi is actually very important. The Golgi is like a packaging factory where proteins are kind of labeled up so that they can be shipped out to different parts of the cell, or maybe even out of the cell if it's a protein that's meant to be sent out of the cell, right, or some other molecule. And how does it do that? So, not surprisingly, the Golgi has to work as a partner with the rough ER. If the rough ER is where these proteins are being made, then they got to be sent to the Golgi so they can be packaged, right? It's the packaging part of this factory, and then sent out. So the Golgi is like a flattened stack of pancakes. One side of that stack of pancakes would be closely pressed up against the rough ER so that proteins that are coming out of the ER can be received by the Golgi network, right? And so that side or face of the Golgi pancake stack is what we call the cis-Golgi network, right? It faces the ER, and therefore proteins that are coming from the ER in little vesicles, this is where the vesicles would arrive at the Golgi. The other side of the Golgi, where you would have vesicles leaving, kind of shipping product, just like trucks at a big warehouse, right, manufacturing plant that then are loaded up with product to then hit the highways and go to different distribution sites, right? That would be the trans face of the Golgi, or the trans Golgi network. Right? Here you can see this is the Golgi over here. Here you see the nuclear envelope, right? So here's the nucleus. You don't really see the ER pictured very well over here, but you can see, you get the impression, right, that these different things are closely sort of coordinated. This cartoon does a better job of showing it, I think. Right. So here you have the nucleus. You would have, the nucleus is actually continuous with the membranes of the rough ER over here. All these little blue dots are the ribosomes, right? And all the tiny little dots are the finished proteins that are being made inside the rough ER. When they're ready to be moved to the Golgi, parts of the endoplasmic reticulum bud off, right? That's where the vesicles come from that are transporting things. And so the vesicles then move to the cis Golgi network. So you can really see this is like a stack of pancakes over here. And inside the Golgi network, they will then be further modified, right, with different tags, molecular tags that identify different destinations inside of the cell. So by the time they reach the trans face of the Golgi and they start budding off as vesicles, right, different signals, different proteins that are part of the membranes of these vesicles that are budding off end up doing things like sending them out of the cell, or maybe just transporting them somewhere else in the cell. So from left to right, proteins that are being made in the rough ER moved through the Golgi and eventually somewhere else. This is what we call anterograde transport. Stuff sort of emanating originally from the nucleus heading out, right? That's anterograde. But things can also move in the opposite direction. Think about raw materials like nutrients that are being taken in by the cell, right, through processes like endocytosis, right? How do those things, when they're inside of a vesicle, how do they know where they need to go in order for your cell to be able to actually use those raw materials, right? Just like we talked about the manufacturing plant, the factory, just as you have on one end trucks shipping finished product out, you must have another part where trucks are delivering raw materials, right? So that's what the Golgi also handles. So these are incoming, right, or retrograde movement of vesicles that go through the Golgi, sort of going backwards in a way, right, that can get labeled up and sent to different parts where these raw materials can be used, right? For example, the ER. All right. So what are these labels that I'm talking about? Right? How does the Golgi package things up? A lot of times it has to do with glycosylation. So glycosylation meaning the addition, the post-translational modification to a protein of sugar chains, right? Small polysaccharide or oligosaccharide, because polysaccharide usually means something really long. Oligosaccharides means a short chain of sugars. And so these get added on in the ER and also in the Golgi, and they serve as little identification tags of where molecules need to go. So here you have an example. We have certain proteins that receive the sugar oligosaccharides. You can see here's a typical one, a sugar oligosaccharide. It's not just one chain. It can be this somewhat complicated branching chain of sugars that involve things like these blue squares meant to be N-acetyl glucoses, right? And then you have these green ones. These are mannose sugars, right? Which are another example of a six-ringed sugar like glucose, right? So you have these mannoses, right? By adding these types of flags or signals on here, you can imagine, right? They not only potentially change the shape of the protein that they are attached to, but they also can represent certain shapes that other proteins are meant to recognize. So those other proteins can dock here and move things around. Why is that relevant? Well, let's talk about COVID again, right? I think all of you know the virus, the SARS-CoV-2, the way it attaches to the surface of your epithelial cells is that it has these spike proteins on their surface, right? And these spike proteins are the right shape to be able to fit and interact with certain proteins on the surface of your body cells, like the ACE2 receptor, right? We now know that the glycoprotein spike is a glycosylated protein structure. It has certain types of these mannose sugar oligosaccharides attached to it that actually help determine its correct shape to interact with your host cell, right? So there's a lot of research now in understanding what these different mannose sugar oligosaccharide flags are that get put on the glycoprotein spike in order to get it into the right shape to be dangerous, right? Okay, in terms of infecting our cells. So oligosaccharides that are added to asparagine residues, right, capital N is asparagine, in the protein target over here, we call this type of glycosylation N-glycosylation, right? N because it's asparagine residue that gets this oligosaccharide attached to it. Serines and threonines can also get certain oligosaccharides attached to them. If that happens, we call this process O-glycosylation, right? So sugars are added to the serines or threonines. So this slide hopefully summarizes this so that you understand it better. So let's say in the rough ER, right, you have all these little green ribosomes on the outside of the rough ER, busy sort of finishing the translation of their different protein products in there. So let's say you have a particular protein that's made that's an enzyme, right? So that's this purple sphere here. Right? In the rough ER, already certain types of carbohydrates are added to it that help it be transported in vesicles to the cis face of the Golgi network, right? This face of that pancake stack, that's the Golgi. Right? Once inside the Golgi, it's going to now be sort of packaged up for transport to wherever it needs to go. Let's pretend that this particular protein is meant to be a protein that functions in your cells as part of lysosomal vesicles, right? In other words, in order for this protein to be useful, it has to end up inside of lysosomal vesicles in your cell, right? Not in the nucleus, not in a mitochondria, and certainly not secreted by your cell because then it would just be lost. So the question is, how does, right, this kind of trafficking happen within the cell to get it where it needs to be? In the Golgi, you end up adding, the Golgi ends up adding things like phosphate groups to it. All right, so this branching carbohydrate chain, let's pretend that this is what we're looking at. So here's the protein that's made in the middle, right? So the blue things are like the amino acid sequence here. And so it's in this ball, right, this purple ball. And certain residues, amino acid residues, get these mannose oligosaccharides stuck to them as little flags or little labels. Right? If we were to zoom in, you'd realize that each one of these little gray balls here, right, is a mannose sugar. So here's the mannose sugar. And so when a phosphate group gets added to it in the Golgi, right, you start having little phosphate groups added to these, and that serves now as a sort of, what is it, label or shipping sticker or something like that, right? So as it moves through the Golgi network towards the trans side, what you'll find is the membrane of this trans Golgi network will have all kinds of proteins as part of it. Right? Certain regions of it might have denser numbers of certain receptor proteins like this blue one in the wall of the Golgi. And this blue one happens to be a receptor that is the right shape to bind to phosphorylated mannose sugars. So here's the receptor protein that's in the wall of the Golgi, right? And you can see here's the chain of mannose sugars, the last three that are part of this particular big branching thing. And here's a phosphate group. Oh, sorry, here's a phosphate group, right? This little orange thing is the phosphate here, right? And this mannose chain is attached to our purple enzyme. Yeah, this is kind of a little out of scale, right? Because in reality, this purple ball here is just one amino acid of a big chain of amino acids, right? That makes the whole big purple ball that's actually being shown in the cartoon on the left here. Okay, I think you guys could probably figure that out, but I'd clarify. So why is that important? Because that means this lysosomal enzyme then, as it's being labeled, it will tend to collect in these regions of the trans Golgi where these receptors are binding to them and holding them to the inside of the wall. This region of the Golgi membrane then, when it buds off, it has other proteins in it as well in the wall, right, that interact with proteins that you'd find in the wall of the lysosome vesicle. So as a result, this vesicle that buds off, this is the little truck now that's hitting the highway, it's carrying cargo, it will preferentially bring that cargo to the lysosome, right? Where then this protein can then be released inside of the lysosome. It will then fall off of the receptor, and now this protein can go to work, right? So you would have similar idea of these vesicles that are meant to, maybe they would have proteins on their wall that maybe associate with proteins on the wall of the cell membrane, the outer cell membrane. So any proteins that preferentially get loaded into these vesicles will end up getting shipped off to the cell membrane, where they might then, the contents, the cargo, might be released out of the cell, right? Amazing, right? When you think about it, that this is all a function of these molecules just interacting with each other. All right, so that's all the new material for the exam, all right? But I thought I'd finish today with sort of a fun discussion, since we have a few more minutes. We've been discussing how important it is that in order for cells to be alive and carry out all of their functions, you have to have membranes that define certain spaces, the cell itself, as well as inside the cell, all these spaces where proteins can be concentrated, different raw materials, different types of other macromolecules, right, can be concentrated in order for specific types of functions to be carried out. But then again, you think about it. All right, so the membranes need to be there in order for the cell to be able to carry out these functions. But then again, the membrane itself is built up by proteins, little machines, constantly making membrane, using fatty acids and things and putting together, right, these different specialized membranes. So you get into a weird sort of chicken and the egg kind of thing, right? Obviously, you need to have a genome that has all the instructions for making things like proteins, right? And the proteins then know how to make the membranes, but yet the genetic instructions, the genetic material won't work if you don't have membranes around them to start with, right? So how did the first cells then do this? What came first, cell membranes or the genome? Right? About 4 billion years ago, how did these things come together in a way that would actually work? Well, nucleic acids, we now realize, can pretty much spontaneously come together in different types of structures just because of the nitrogenous bases of nucleotides interacting, right? So for example, we already learned that the fact that uracil can interact with adenine or cytosines can interact with guanines through hydrogen bonding, that's a critical feature of how our DNA is organized, right? And how DNA and RNA can associate with each other. Here you have a couple other nitrogenous bases. Oh, here's that barbituric acid again that we discussed, right? And it's a nice reminder, yeah, barbituric acid is just another example of a nitrogenous base. Here's another one, melamine, right? And you say, wait a minute, we've always just been talking about like 4 or 5 of them, the A's, the C's, the G's, or the U's in RNA, and then we have T in DNA, right? So in reality, though, this is a reminder that there are like an almost infinite number of different examples of small nitrogenous bases. It's just barbituric acid and melamine, right, are not used by life on Earth as the basis of genetic material, but we certainly have it here on Earth, right? Melamine, in fact, is used as an additive, right, synthetic additive for certain types of dairy products, so that the perceived dairy content or protein content of something like, cheaply processed milk looks like it's a lot higher quality. Right? So here you have barbituric acid just coming together to form a nucleotide. Here's a ribose ring, phosphate group. Hey, this is a nucleotide. It's just not a nucleotide that our cells would naturally use. Right? So these things can come together like a melamine nucleotide, barbituric acid nucleotide, and they would form just more complicated structures, right? And already you can tell that the fact that nucleotides come together to form things like nucleic acids, right, could just happen anywhere, right? It doesn't really require necessarily proteins to have gotten this process started. Okay. So if our genome more or less could start coming together just by nucleotides coming together spontaneously, right? But what about this whole issue of the fatty acids that make up our cell membranes? Right? This is a neat piece of evidence here. This is a rock, a really old rock, like 3.9 billion years old, right? Where they've zoomed in really close using a super high electron microscope. And what they find are these funny little fossilized structures, which are actually lipid bilayer bubbles, right? These are bilayers of phospholipids that have come together to form little bubbles, right? So if this existed 3.9 billion years ago, right, you might say, oh, so that makes sense then. If nucleic acids can just spontaneously form by nucleotides coming together, right? And we had these, phospholipid little bubbles on Earth already, then those are your two basic ingredients to get something like a cell started, right? But the real kicker is, right, where did these phospholipid bubbles come from, right? So the neat part about this is this rock, right, is not just any old ordinary rock that they're looking at where they see these things. It is a meteorite, right? So meteorites raining down on Earth bring raw ingredients, right? Things like amino acids, DNA bases, sugars, fatty acids, right, actually came over the course of Earth's early development from outer space, raining down from other parts of our galaxy and other parts of the universe, right? And that's how life was seeded on our planet 4 billion years ago. Certain things could spontaneously come together, right? But certain things, right, were actually helped out by being brought from the skies, raining down, right? So that's why our story of life on Earth doesn't really begin with things just starting off, with raw materials being on Earth itself, right? Because the raw materials that make up Earth are no different than the raw materials that make up other planets throughout the galaxy and the universe, right? So as asteroids, meteorites are crashing on different planets, these same organic compounds were seeded, right? And it's just on our planet, because of our liquid water, that set the stage for how these different compounds could come together in a solvent, water, that allowed them to start coming together to form life, right? So for those of you who sometimes get mad at your partner or significant other, and you always say, sometimes I think he or she's from another planet, right? Well, you're partially true, right? But that's true of all of us and all life on Earth, which is why I think it's wonderful to think about this, right? That these raw ingredients are crashing down on planets in other places as well, other galaxies, other corners of the universe, right? But the fact that they came together in this certain way on our planet is because of liquid water. How are they coming together on other planets that might have other solvents other than water, right, in abundance on their surface? So that's something to think about. All right, okay, let's go ahead and stop there.